Hello students, welcome to Zoology Classroom. Myself Yal Mahesh, Professor of Zoology, Tara Government Degree College, Sangareddy. In today's class, we are going to discuss about absorption of digested food materials. So till previous last classes, we have seen that how the food materials, different types of food materials are digested in our digestive system. So when all the food materials, when they reach the small intestine, almost all types of food materials will be digested. The digested food materials will be absorbed into the bloodstream in the small intestine. Here in this slide, we are seeing a section of small intestine. The inner side of the small intestine, it is having many finger-like projections into the lumen of the small intestine. These finger-like projections are called as microvilli. So it is through these microvilli only the digested food materials will be absorbed into the bloodstream. So in this slide, we are seeing a single microvilli. So each microvilli, it is bordered by a single line of cells. Single line of cells are there on the outside of this microvilli. So, and again, these microvilli also, the cells on this microvilli also, further they are having many brush borders on the uh, side which is facing towards the lumen of the small intestine. So in this way, small intestine is having many finger-like projections called as microvilli. And microvilli also, it is bordered by a single layer of cells. And the cells towards the lumen side, they are having a brush border. And this brush border of the cells and finger-like projections are microvilli of the small intestine. All these structures, they are increasing the surface area. When the surface area is increased, the absorption capacity also will be increased. So the presence of these microvilli and presence of brush border on the cells which are bordering the microvilli is to increase the surface area or absorption area. So in this slide, we can see how the digested carbohydrate materials are absorbed into the bloodstream. So if you carefully observe this slide, we have to understand that here this box-like structure, okay, this box-like structure, it is the brush border, the cell having a brush border, or it is the cell bordering the microvilli of the small intestine. And towards this lumen side, this brush border actually is having these the cell it is having brush border towards this lumen side. Okay, actually this cell is having a brush border like this. And this area is the lumen, and this area is the uh, wall of the small intestine. In the walls of the small intestines or in the walls of these microvilli, many blood vessels will be there. So, and this cell, this box-like structure, this is nothing but this cell which is having brush border, and this is the border of the microvilli. So, till now, from this, okay, this area, this total area is the wall of the small intestine. And this area is the lumen of the small intestine. So in this lumen of the small intestine, okay, in this lumen of the small intestine, we are having digested food material. So how these digested food materials are taken into the bloodstream? If the blood materials, if the digested food materials are about to enter into the bloodstream, first of all, they have to enter into the cells which are having this brush border. So how the digested food materials are entering into the cells, border cells, and from there how they are entering into the bloodstream. Let us see. So here, after the digestion of carbohydrates, we will have different types of monosaccharides. And of these monosaccharides, glucose, GLU, it stands for glucose. Glucose will be there, and fructose also will be there. And apart from this glucose and fructose, galactose also will be there. Okay, and some disaccharides also like maltose. Okay, some disaccharides like maltose also will be there, and lactose also will be there. So this lactose after the digestion with the help of lactase enzyme, this lactose molecule will be digested into one glucose molecule and one galactose molecule. Glucose molecule and GAL, it stands for galactose molecule. And maltose, okay, disaccharide maltose with the help of maltase enzyme, it will be digested into two glucose molecules, GLU plus GLU. So one maltose will be digested into two glucose molecules. And whereas the sucrose, it will be digested into glucose and fructose. So if we carefully observe this diagram, we can come to know that this is the cell bordering this microvilli, and the cell membrane of this border cell, it is having lactase enzyme and maltase enzyme. So lactase enzyme and maltase enzymes are integral parts of the uh, plasma membrane of this border cell. So lactose and maltose will be digested once they are attached to these enzymes, lactase enzyme and maltase enzyme. And this lactase enzyme and maltase enzyme, they are attached to the plasma membrane of this border cell. Whereas this sucrose molecule will be digested by the sucrase enzyme, and sucrase enzyme is not attached to the plasma membrane of this border cell. So sucrase enzyme is free in the lumen. So it will be released into the lumen. So where in the lumen, this sucrase enzyme digests the sucrose molecule into one glucose and one fructose molecule. So as a result of this digestion of all polysaccharides and disaccharides, okay, as a result of all polysaccharides and disaccharides, finally we will have glucose molecules, galactose molecules, and fructose molecules. So these are the three main types of monosaccharides will be there. So let us see now how these three monosaccharides, they are entering into this border cell, and from there how they are entering into the bloodstream. So these glucose and galactose molecules, okay, glucose and galactose molecules, they cannot enter into the border cell on their own. Okay, they cannot enter into the border cell on their own. They need the assistance of some other molecule, that is sodium ion, Na plus. This is Na plus sodium ion. So on the plasma membrane of this border cell, there is a separate, a special transporter molecule or a special transporter protein is present, that is called as SGLT, sodium glucose transporter. Sodium and glucose transporter. So the sodium glucose transporter is transporting a glucose molecule along with sodium molecule. Otherwise, it can also transport one galactose molecule along with sodium molecule. So glucose molecules and galactose molecules will be transported from the lumen into the border cell cytoplasm. Okay, along with sodium only. When sodium is not present in the lumen, these cannot enter into the cytoplasm of this border cell. That means when sodium is absent in the digested food material, that means if we are lacking sodium in our food, even though the carbohydrates are digested, even though plenty of glucose and galactose molecules are available in the digested food material, they cannot enter into our cells. They cannot enter into our body. Thus, we will lose them. So in order to get them into our body, we need to have sodium in our food. So along with sodium only, these glucose molecules and galactose molecules, they will enter into the cell. So after entering into the cell cytoplasm of this border cell, this glucose and galactose molecules, okay, this glucose and galactose molecules, again, they leave the cytoplasm and they will enter into the bloodstream. So this pipe-like structure, it is representing the blood vessel, especially hepatic portal veins. So all the hepatic portal veins, all the blood vessels coming from the small intestine, they will join together to form a big blood vessel. This blood vessel carries blood from this intestine to the liver. So this area is the liver. So all the blood coming from the small intestine region will be supplied to the liver through this hepatic portal vein. Hepatic portal vein. So the glucose molecules and galactose molecules, after entering into the cytoplasm of this border cell, they will directly across the concentration gradient, they will directly go out of the cytoplasm and they will enter into the bloodstream. So here the sodium is left behind in the cytoplasm only. Again, this sodium, it has to be pumped back into the lumen. Why? Because there are more glucose molecules and more galactose molecules there out in the lumen. So in order to bring them back into the cytoplasm of the cell, sodium has to again go out of the cell into the lumen. So for that purpose, to send the sodiums out of the cell into the lumen, ATP energy will be utilized. So in this way, glucose and galactose molecules are absorbed into the cytoplasm of the border cell at the expense of ATP molecule. So it is called as active transport. So in order to absorb sodium and in order to absorb glucose and galactose, our cells, our border cell, they will spend some energy in the form of ATP. So without ATP expenditure, so glucose and galactose cannot enter into the cell. Why? Because sodium has to come out of the cells. If it has to come out of the cells, ATP energy has to be ATP energy has to be utilized. Then only sodium comes out and brings some more glucose and galactose into the cell. So in this way, glucose and galactose molecules are absorbed by active transport method coupled with sodium ion. So they are coupled with sodium ion and by utilizing ATP energy only, they are entering into the cell. And after entering into the border cells without ATP energy, they are going into the bloodstream. And from bloodstream, they go to liver, and from liver, they go to heart, and from heart again, they are pumped to all body parts in our body. And coming to another thing that is fructose. Okay, next one is fructose. Fructose directly it transported into the cell with a special transporter called GLUT5 transporter. So GLUT5 transporter sends fructose into the cell directly. For this purpose, ATP energy is not required. So fructose is transported into the cytoplasm by a passive transport method. Whereas glucose and galactose molecules, they require ATP energy in order to absorb them into the cells. So in this way, after fructose entering into this cell, it goes out of the cell and reaches the bloodstream in the hepatic portal system. And this transporting glucose, galactose, and fructose from the cell into the bloodstream, a special type of transporter, GLUT2 transporter. So GLUT2 transporters, they will send this glucose, galactose, and fructose molecule from the cells into this bloodstream. So in this way, GLUT2 is helping in the passive transport of glucose and galactose and fructose into the blood. So this is about this uh, absorption of carbohydrates. Next, in this slide, we can see how the amino acids are absorbed into the our body or our blood system. So, like in the previous slide only, here you just understand that this box this box is the border cell, and this is the inside of this border cell or the wall of the small intestine where many blood vessels and other things are present. And this is the outside of the border cell or lumen of the small intestine. In this lumen only we are having digested food materials passing through this lumen. So the digested food material in the lumen, it is having many fully digested amino acids and some partially digested or incompletely digested dipeptides and tripeptides. Some tripeptides will be there. As a result of this proteins digestion, mostly amino acids will be there. Apart from these amino acids, some dipeptides here, it is depicted with the two dots or two balls joined together with a link. And here, this is a tripeptide in which three amino acids are represented by three ball-like structures which are connected with two peptide bonds. So some tripeptides, some dipeptides, and many amino acids, free amino acids will be there as a result of this digestion of protein. So how these free amino acids, some tripeptides and some dipeptides, how they are entering into the border cell cytoplasm, let us see. So on the plasma membrane of this border cell, some dipeptidase enzymes are there. So these dipeptidase enzymes, they will digest the dipeptides into free amino acids. And these free amino acids, they are transported into the cell coupled with hydrogen ions. So without the presence of hydrogen ions or without the help of hydrogen ions, amino acids and dipeptides and tripeptides, they cannot enter into the cell. So in order to enter into the cell, the free amino acids and dipeptides and tripeptides, they need the help of hydrogen ions. So these hydrogen ions are coupled to these amino acids, dipeptides and tripeptides. Like this, these free amino acids, see these free amino acids, tripeptides and dipeptides, they enter into the cytoplasm of this border cell. After entering into the cell, again these hydrogen ions, which were coupled to these free amino acids, dipeptides and tripeptides, they will, again these hydrogen ions, they will be pumped back into the lumen in order to bring some more amino acids, dipeptides and tripeptides into the cell. So to send these hydrogen ions across the concentration gradient into this thing, some energy, ATP energy will be utilized. So in this way, amino acids and dipeptides and tripeptides are absorbed at the expense of some ATPs. So they are being absorbed by active transport method. So they are being utilized some ATP energy, then only they are able to enter into the cytoplasm of this border cell. And not only hydrogen ions, sodium ions are also helping in the transportation of this amino acid. So some free amino acids coupled with sodium ion, some special transporters are there which are transporting these amino acids coupled with sodium ions into the cytoplasm. So some free amino acids are then are being pumped or absorbed into the cell coupled with hydrogen ions, and some more free amino acids coupled with sodium ions are also absorbed into the cytoplasm. And again these sodium ions, okay, again these sodium ions, they will be utilized in some more different processes where sodium ions are pumped back into this bloodstream and are out of the cell, and sodium ions are sent into this thing. So that is a different story. So in this way, amino acids and dipeptides and tripeptides are absorbed into the body, into our body, at the expense of ATPs only. So these are coupled with hydrogen ions and sodium ions in order to absorb into the cell. So once they are entering into this thing, all free amino acids, they will enter into this hepatic portal system. From there, from this hepatic portal system, they will go to liver, from liver to heart, and there, from there, to all body parts, they will be supplied. So, and what about this, some dipeptides and tripeptides? They entered into the cytoplasm. So, in the cytoplasm of this border cell, peptidase enzymes are also there, dipeptidases and tripeptidase enzymes. So, these dipeptidase enzymes and tripeptidase enzymes, they will digest these dipeptides and tripeptides completely and finally convert them into free amino acids. So even though some dipeptides and tripeptides, they enter as is into this cytoplasm, once they enter into the cytoplasm, they will be completely converted into free amino acids. So finally, free amino acids only, they are going into the bloodstream in the hepatic portal system. So in this way, this uh, digested proteins are being absorbed and sent into the blood. So in this slide, we can see how the absorbed fatty acids or fat materials, how the digested fat materials or fatty acids are absorbed into the bloodstream. So like previous slides, here also we have to understand that this box-like thing, it is a border cell, and this is inside of the intestine, and this is the lumen of the intestine. So in this lumen of the intestine, we are having digested fatty materials. So as a result of complete digestion of fat materials, what we will get some bile salts and we will get some volatile fatty acids, and we will get some cholesterol, vitamins A, D, E, K, vitamin A, vitamin D, vitamin E, vitamin K, and free fatty acids, cholesterols, and some esters. So all those things will be present in the digested material. So all these things, bile salts, okay, all these bile salts, they will be absorbed into the bloodstream and they will be sent to this liver again to form the bile juice. We know that in the bile juice, sodium glycopolate, sodium taropolate will be there. So in order to prepare sodium glycopolate, sodium taropolate, these bile salts have to be reabsorbed. So again, these sodium glycopolate, sodium taropolate, when they are released into this uh, duodenum, finally after the digestion is completed, they will be converted into bile salts. So in this way, bile salts are being recycled forever in our body. So bile salts, mostly bile salts are not excreted. In some rare cases only, whenever they are excess salts, then only they will be excreted through the fecal matter. But mostly they are reabsorbed into the bloodstream and they will be recycled. So coming to this remaining materials apart from this bile salts, what's happening in the lumen of the small intestine? Vitamin A, vitamin D, vitamin E, vitamin K. So all these vitamins, they are forming some globule-like structures. So these globule-like structures are called as micelles. So these micelles are also having some free fatty acids. Some free fatty acids are also joined together with all these vitamins. So vitamin A, D, E, K, and free fatty acids and cholesterol. So all those things, they will join together to form some globule-like structures, droplet-like structures. So these droplet-like structures are called as micelles. So these micelles will be absorbed into the cytoplasm of this border cell. So these micelles, they will be absorbed in a passive manner. So they are following a passive transport method. Why? Because we know that plasma membranes are made up of a phospholipid bilayer. So and these micelles are also, they are nothing but they are also lipids. So as they are lipids, easily this phospholipid bilayer gives way for these micelles to penetrate through them. So that's why micelles, and for that matter, for any fat soluble material, energy is not required to go across through these plasma membranes or any biological membrane. So without ATP energy, simply passively these micelles, they will enter into this cytoplasm of this border cell. So once they enter into the cytoplasm of this border cell, micelle will be broken down into cholesterol, vitamin A, D, E, K, free fatty acids, and monoacyl glycerols and glycerols. So all these things, they will be absorbed into this smooth endoplasmic reticulum. So once they enter into the smooth endoplasmic reticulum, all these things, they will be processed, and from these things, processed materials will finally, they will enter into the rough endoplasmic reticulum. So after they enter into the rough endoplasmic reticulum, finally they will be converted into chylomicrons. So they will take the form of chylomicrons. Again, these chylomicrons, they will leave this border cell and they will enter into the lacteal. So we know that in each finger-like structure, that microvilli. So in each microvilli, there is a single lacteal duct present in them. So this lacteal duct absorbs all these chylomicrons and other fat-soluble material. So these chylomicrons, they will enter into these lacteals, and from these lacteals, they will enter into the lymphatic system, lymph system, and from this lymph system, it will transport these chylomicrons or fat materials to the skeletal muscles. In the skeletal muscles, they will be stored. The skeletal tissues, they will be stored in the form of LDL, low density lipids, like that. And some of these chylomicrons or some of these fat materials, they will be also stored in the adipose tissue. So like this digested materials, all these digested fat materials, they will be absorbed in the form of micelles, and in the brush border cells, they are converted into chylomicrons. Chylomicrons are transported into the lacteals. Some of the chylomicrons may also enter into the bloodstream, also into the uh, hepatic portal system, but mostly chylomicrons, they will enter into this lacteal. From lacteals, they will enter into the lymphatic system. Through lymphatic system, these fat materials, they will be sent to the skeletal muscles and adipose tissues, where these fat materials will be stored in these two things, adipose tissues, skeletal muscles. And excess of these materials from this thing, again, they will be sent to this liver. From liver, they will be sent to this heart through the blood circulatory system. So this is how digested fat materials are absorbed and circulated throughout our body. So next, how the digested nucleic acids are absorbed into this thing. So as a result of digestion of nucleic acid, we will get different types of nitrogenous bases. We know that nitrogenous bases are four types in DNA: adenine, guanine, thymine, cytosine. And whereas in case of RNA, we will get a next different one, uracil also there. So totally five types of nitrogenous bases will be there: adenine, guanine, thymine, cytosine, and uracil. And pentose sugars will be there. They could be ribo sugar or deoxyribo sugar, and phosphate ions are also will be there. So all these things are also, they also will be absorbed into this border cell through active transport method. And water absorption. So daily around 9 liters per day water enters into the duodenum, and 90% of water is absorbed into the small intestine by osmosis process. So water absorption takes place by osmosis process. Through this osmosis process, water travels from a low concentration region to high concentration region. So coupled to active transport of sodium and other solutes also. So sodium transportation also helps in the water absorption process. So water moves in both direction across the intestinal mucosa. This is the very peculiar feature of water transportation. Why? Because whenever our intestinal juices are in intestinal wall, membranes are having high water content, water enters into the lumen. That means water leaves our body, and that leads to dehydration process. So that's why it is very dangerous situation when a patient is severely suffering from this loose motions or diarrhea like that. So when diarrhea is there, water leaves our body, water leaves from our tissues. That leads to a very dangerous critical situation. That is because this water peculiar feature. Water can move in both directions. Water not only enters into our body through this lumen, water leaves the body also and enters the lumen also in some special cases when this type of pathological conditions prevail. And absorption of vitamins. So in small intestine fat soluble vitamins A, D, E, K, they will be carried in the form of micelles. This thing we have already seen. So they need not any ATP energy. Simply passively they will enter into the cells in our body. And water soluble vitamins C vitamin and B vitamin, they will be absorbed by diffusion process or passive transport, and in case of some vitamins active transport also take place. And vitamin B12, particularly this B12 is a very large and charged molecule. It binds with intrinsic factor. It is absorbed by endocytosis. This is a peculiar feature of this vitamin B12 absorption. And in large intestine, vitamin K and vitamin B vitamins are also absorbed. Why? Because in this large intestine, some bacteria, symbiotic bacteria are living there, and these symbiotic bacteria, they will produce this vitamin K. That's why vitamin K will be absorbed in this large intestine region only. And coming to the electrolytes absorption, and electrolytes will be composed of sodium ions, potassium ions, iron ions, and calcium ions. So how these ions are absorbed? Let us see. Sodium ions, they are coupled with the glucose and amino acids. Okay, we have already seen about this thing. So sodium will be absorbed by active transport method. And potassium facilitated diffusion or paracellularly. So sodium will be absorbed into the cell with the help coupled with sodium. So when sodium leaves the cell, potassium enters into the cell. When potassium leaves the cell, sodium enters into the cell. So both these things, they will travel vice versa into and out of the cells. This is called as facilitated diffusion and depends on concentration gradient set up by water absorption also. This potassium will be absorbed into our blood stream. And diarrhea creates potassium balance problems. As I have already told, during diarrhea conditions, water leaves our body and goes to outside through this diarrhea. So during that conditions, a lot of potassium leaves our body and goes out of the body in the form of motions. That leads to a lot of potassium balance problems and neurological problems and buffering problems also will arise due to this condition. And iron ions, they are stored in mucosal cells. And these mucosal cells, they have a special protein called ferritin on the plasma membranes of them. And this iron will be bound to this ferritin and it will be stored in these cells. And mucosal iron barrier and 4 times more in women than in men. So this ferritin is 4 times more present in women than in men. So only released when needed or lost as mucosal cells slow. So this iron is bound to these ferritin proteins. Whenever there is a necessity, then only these iron ions are released into bloodstream. Otherwise, they will not be released. Next, calcium absorption. Calcium absorption also made possible only when blood calcium levels are decreased. So when the blood calcium levels are very low, then only calcium will be absorbed from this digested food materials. And calcium absorption also, it is regulated by vitamin D and parathyroid hormone also. So this is about this absorption of digested food materials. With this, today's session is over. We will meet in next